The polymerase chain reaction, or PCR, is a powerful method for obtaining millions of duplicates of a DNA fragment from just one original copy. The reaction begins with a solution containing the DNA from a cell. The goal is to amplify a specific region, the target DNA. The solution contains Tack Polymerase. Tack is a type of heat-stable DNA polymerase derived from a species of bacteria living in hot springs. Because Tack Polymerase continues to function normally at high temperatures, using it allows researchers to separate the DNA strands using heat without destroying the polymerase. The solution also contains primers, short single-stranded DNA molecules, and plenty of deoxynucleoside triphosphates, or DNTPs, which are not shown at this scale. Let's examine the basic PCR cycle, which is typically repeated about 35 times. Temperature changes are automated by PCR machines. In the first step, denaturation, the solution is first heated to nearly boiling, 95 degrees Celsius. The heat breaks the hydrogen bonds between the two DNA strands and allows them to separate. Despite the intense heat, the Tack polymerase remains active. In the second step, primer annealing, the temperature is dropped to around 60 degrees Celsius. The exact temperature depends on the length and base composition of the primers. At this relatively low temperature, the primers can form hydrogen bonds with the single-stranded DNA. Two primer types are created, each one complementary in sequence to one of the two ends of the target DNA. To make the primers, the sequences at the ends of the target DNA must be known. In the third and final step, extension, the temperature is increased to 72 degrees Celsius. This is the optimal temperature at which Tack polymerase functions. The primers are essential in this process because they provide free 3' hydroxyl groups to which the polymerase can add additional DNTPs. Each new DNTP that joins the growing strand is complementary to the nucleotide in the opposite strand. At the end of this first cycle, there are 2 DNA copies instead of the one original copy. In cycle 2, the process repeats so that there are now 4 double-stranded copies. The process continues, doubling the number of target DNA copies with each cycle. Doubling occurs because each newly synthesized segment of DNA serves as a template in the subsequent cycle. Notice that more and more of the fragments consist of just the target DNA. Roll your cursor over the histogram bars in the graph to find out how many target DNA molecules are produced in each cycle. You can see how the exponential increase in copies with each cycle results in an enormous number of copies by the end of the 35th cycle.